We would like to thank you for accepting our invitation for this interview and sharing a bit of your time with us. In 1982, you were one of the researchers to discover the first human oncogene, RAS, which makes normal cells start to present tumoral phenotypes. Could you briefly tell us about the process and how you were able to make this discovery?
Well, in the mid-1970s, we were thinking about how cancer is formed. The prevailing notion was that cancer cells are mutant cells, but in fact there was no experimental proof that cancer cells had mutant genes in them. And so that began to provoke a search for chemically transformed cells that might carry a mutant gene in them that might be responsible for the altered behavior of these chemically transformed cells. These cells had no tumor viruses in them, and until that time, tumor viruses had been the prime object of research in cancer biology at the molecular level. And so we developed a technique that's termed transfection that allowed us to introduce normal DNA or cancer DNA into normal cells. That is, we could transfer the genes into them. And so we undertook a series of experiments starting in 1978, 79, that allowed us to prove that DNA from chemically transformed cells actually carried some kind of genetic element that could, when introduced into normal cells, this cancer cell DNA could cause the normal cells to become neoplastic, to become cancerous. And this demonstrated that the DNA of a cancer cell actually carried genetic information, ostensibly mutant genes, that was responsible for the altered behavior of those cells. 3 years later, we and others isolated the responsible gene. And as we and others found out, the responsible gene was a RAS oncogene that had been discovered in the context of retrovirus virology some years before. And so this demonstrated that a normal cellular gene called RAS could be mutated into a cancer gene through actually a very small, subtle mutation, a point mutation in the normal gene. That was the order of experiments that we undertook.
So it was amazing for us to read at the textbook that you described the process, how the oncogene is formed. It's really interesting.
I'm pleased to hear that.
You were a co-author of the article The Hallmarks of Cancer in the year 2000,
Yeah.
which later underwent some updates in 2011 that gave rise to a new edition called Hallmarks of Cancer Next Generation. How did the idea of writing the Hallmarks of Cancer arise? Did either you or your colleagues have any clue of the proportion this article would gain in the international scientific community?
These are all very good questions. By 1990s, it became clear that cancer research was getting more and more complicated. And no one could figure out whether there were small numbers of underlying principles that unified most kinds of cancers. In fact, Doug Hanahan and I were both graduates of MIT, where we learned mathematics and physics. And learning physics, we realized, of course, that Newton's laws of mechanics could unify much of the physical universe to a simple set of laws. But biology was getting more and more complicated and seems to have had no underlying common principles. And so in the year 1999, Doug Hanahan and I attended a cancer research meeting in Hawaii. And during one of the sessions, we played hooky. That is, we ran away. He had rented a car, and we drove to the top of a volcano to see what it looked like. An extinct volcano, by the way. And while we were going there, we were talking about the complexity of science, specifically the complexity of cancer research, which was thousands of unrelated phenomena. And we began to converge on the notion that there must be a small number of underlying principles, commonalities that are shared by different kinds of cancer. And so by the time we drove down from the volcano, we agreed, in effect, to write an article describing how we imagined that these cancer traits might be shared in common by various kinds of human cancer. Important is not how these cancer cells, how these cancer traits arise, the mechanisms. What was important about the hallmarks of cancer were the traits, the characteristics, the phenotypes of cancer cells. That's what we wish to describe.
Yeah.
And so in the autumn of 1999, I got an email from Cell saying, would you like to contribute a review article to the January 2000 issue of Cell, which was the first issue of the new millennium. And I wrote to Doug and I said, this seems like a golden opportunity. We may actually find a place to publish this review. Now, I will tell you in total honesty that we fully expected that this review, like almost every other review one writes, would fall like a stone into a quiet pond and disappear deep into the water. No one would ever look at it. No one would ever care about it.
Oh my God.
That was our expectation. And so it was published, and you know the consequences. It was cited many times. It may help cancer researchers rationalize their research. It may help other cancer researchers to avoid having to refer to many papers in the bibliography of the manuscripts that they write. In any case, now you begin to understand what we were thinking about when we started the hallmarks of cancer. Part of my thinking was influenced by what a British cancer researcher once told me in the mid-1980s. He said to me, he was a bit cynical, he said to me, never should you ever confuse cancer research with science. And what he meant was cancer research was only a complex collection of phenomena. It had no underlying principles and no underlying logic, and therefore it was hardly a scientific discipline. So that created a little bit of cynical thinking, both in my mind and that of Doug Hanahan's, and we wished to counter that. We wished to indicate that indeed there was a logic that was central to cancer development.
In 2019, your lab did a lot of research regarding the epitélio-mesênquima transition and its implications in the study of cancer. What are the other branches of research your lab is currently working on?
Well, part of what we're interested in at present, besides the epithelial-mesenchymal transition, is the immunobiology of cancer. For example, we have found that mouse breast cancer cells that are in an epithelial state can be attacked and eliminated by immunotherapy, whereas the genetically identical cells that are more mesenchymal, in a mesenchymal state and created by the EMT program, those cells are quite resistant to being eliminated by immunotherapy. Indeed, if we make mixed tumors carrying small numbers of mesenchymal cells and large numbers of epithelial cells, the mesenchymal minority of cells can protect the tumor as a whole, can protect the great majority of the nearby epithelial neighbors from being eliminated. And we find that very provocative because it may explain why many kinds of human solid tumors are resistant to elimination by checkpoint immunotherapy, one of the kinds of immunotherapy that are currently being undertaken.
Do you have any advice for the current and next generations of scientists?
Well, a couple things. If you go and get a training, you may end up working in a lab. And when you work in that lab, you may do, for example, a PhD thesis, a doctoral thesis. And you'll be working on one topic in great detail and in great depth. But people who focus exclusively on that one topic become blind to what's to the left and blind to what's to the right. They become experts about a very small area of science, and once they leave that small area, they're useless. And that's a great danger. And so while you are a student and while you are a pre-doctoral student, you have to be continually enriching your learning about science by looking and reading other kinds of articles and by talking with people who are experts in other areas and other specialities.
Yeah.
The fact is, if you focus only narrowly on your own area of science for all of your career, you probably won't make very many interesting discoveries, simply because, at least in my own experience, if you're working in one area, many of the innovations, many of the interesting discoveries come from bringing information from another field into the work, into the specialty that we're working on. And so it's good to know multiple areas of contemporary biology, not just cancer research.
Yeah.
Secondly, if you go and become a pre-doctoral student, you should make sure that you work for somebody who is interested in your education, not somebody who's interested as the head of a laboratory in using you as a super technician, as simply an extra pair of hands to get work done cheaply and inexpensively in the laboratory. So that's also very important. And thirdly, you should find out whether you really enjoy the work. If you don't enjoy the work, even though your parents believe you should become a scientist, you shouldn't do it. Because if you don't enjoy what you're doing, you will not be a very good researcher. That's very important. There are many people who go into medicine because their parents wanted them to become a doctor.
Yeah.
And when they discovered what medicine is really about, they discover that they don't really like it that much. And yet many of those people continue to become a doctor because their friends and their relatives are very proud of their being a doctor, even though they themselves hate being a doctor. They don't like it at all. So you won't be a very good researcher unless you find your work interesting and provocative and enjoyable.
And thank you again so much for this interview and for letting us borrow some of your time. It really means to us.
Com prazer from my side. I too say muito obrigado. I wish you all the best, including staying safe, staying healthy.
You
Bye-bye.
too. Yeah. Take good care. Bye-bye.
Bye-bye.